So students, the next topic as per NCERT is the vectors which are required for cloning the genes in the plants and the animals. So how do we transfer it into a plant cell or into an animal cell? So the concept talks about a bacterium in the case of plant cell and a virus in the case of the animal cell. So it talks about that there is an agrobacterium tumefaciens which is able to cause tumor to be formed in the plants it attacks. So this tumor causing gene which is present in this agrobacterium, and in the same manner it talks about that there are retroviruses which can be used in the animals, and retroviruses also cause the cancerous growth. And we learned that if we can convert these pathogens by removing their disease causing ability, then you will start calling it as disarmed pathogen. And now because they will be working as a slave for us, transferring the gene of desire for us, that's why they are called as disarmed pathogen vector, because they will be vector for us and they will not they will lose the property of infection, so they are called as disarmed pathogen vector. So let's understand. The vectors which are required for the plants are basically the natural genetic engineer. So scientists have discovered that there is a bacterium which survives or lives in the soil, that means close to the root system of the plants, and that bacterium is agrobacterium tumefaciens. Be careful with the spelling of tumefaciens. And this bacterium lives around the root system of certain plants, and those plants are basically the broadleaf plants. Now we all know broadleaf plants are the dicot plants. So this bacterium is a pathogen, and this bacterium has a plasmid which is called as TIE plasmid. So TIE stands for tumor inducing plasmid, and this plasmid has a particular portion, like if you remember, I have discussed before, plasmids provide special features to the bacterium. So here the special feature possible is to cause tumor. So that gene or that piece of DNA which is present in this plasmid, which is responsible for tumor induction, is called as tDNA. So this tDNA, the bacterium is able to use this tDNA, and then it will cause tumorous growth in the dicot plants. And then what will happen, the growth will start, uncontrolled growth of the underground basically portion of the stem etc. will start growing, like in the case of turnip, in the case of beetroot. So the underground portion basically of the plant will start growing bigger in size, and then it will become irregular in shape, and we call it as crown gall. This tumor is called as crown gall. So this crown gall formation is because of the agrobacterium tumefaciens. Now imagine for a second, if this tumor causing property is removed somehow, and then what will happen, it will not cause the infection. But now instead of tumor causing, if you cut and attach a foreign DNA into this bacterium, then it will be a successful disarmed pathogen vector, because it won't cause tumor, but it will still carry the capacity to carry the gene into the plant cell, and finally plant cell would become genetically modified plant. So that's why it is called in biology as natural genetic engineer, because it is able to perform this activity. In the same manner, in animals we can use retroviruses. Now retroviruses are able to change normal cells into the cancerous cells as per the NCERT. So in the same manner we are going to talk about this also, that a retrovirus carries RNA, and we all know first of all it will make a complementary DNA, and this complementary DNA will attach with the DNA of the host organism and then take over the machinery. So now if we are able to somehow remove this property of taking over machinery and causing infection, so what will happen now, this virus will become a disarmed. Disarmed means losing the property of infection. So disarmed pathogen, and it will be a vector now for us, because now we can insert any piece of alien DNA or any piece of alien RNA, and then it will make a complementary DNA for us, and then it will be attached to the DNA of the host cell, and the host cell will get a modified genome. So we can call the host organism or host animal as genetically modified organism. So this is called as the concept of disarmed pathogen vector. So tumor inducing plasmid has been modified into a cloning vector which is not pathogenic to plants anymore, but still retains the ability to deliver the genes of our interest into the plant. So this is what science has already done, that modify these microorganism, and they will become a tool for us. And second, similarly, the retroviruses are also used to carry desirable genes into the animal cells. So this oftenly comes in board exam, that if you wish to use a vector, which vector will you use for plant cell and animal cell? So the answer would be the agrobacterium, which has the tumor inducing ability, and then the retrovirus, which has the cancer producing ability. Now remember that once the genetic gene of interest is joined to the suitable vector, it is transferred into the suitable bacterium, or into the bacteria plant cell, or into the animal host cell, and where it will undergo multiplication. So it will start making copies once it has been successfully shifted into bacteria with the help of a virus, or with the help of bacteria into the plant, or with the help of a virus into the animal cell, and finally the production of the desired product will start. So this is how vectors are explained in the NCERT. Let's understand slightly more about the various vectors which are possible.